Hello, everyone. What we're going to talk about in this brief lecture is two important topics which are extremely relevant for the living organisms. The first thing we'll talk about is what makes water such an important molecule for all living organisms. You'll see that all the properties of water that work in these directions, so the thermal capacity, the cohesion, and the water as a solvent, they all come ultimately from the polarity of water as a molecule and its capacity to form two hydrogen bonds. After that, we'll talk about the pH. By pH, what we mean is the logarithmic measurement of the concentrations of protons in a solution, including in aqueous solutions in living organisms. So we'll talk about what it is and how molecules defined as acids and bases affect it. And then we'll talk about why is it important in living organisms? What are the effects of changes in pH in living organisms? And finally, we talk about how living organisms prevent these changes by using buffers. So starting with water here. Water, if you remember from the previous lecture, it's a polar molecule. And the reason why it's polar is because it contains two bonds, two covalent polar bonds between oxygen and hydrogen. Two bonds that are not opposite to each other, but they are at an angle of 104 degrees. And by doing that, they cause basically the accumulation of a partial positive charge on one side of the molecules and a partial negative side on the opposite end. So the charge is polarized within the molecule. The molecule is defined as polar. Now, because of that, water can also form hydrogen bonds. So this positive charge or partial positive charge associated with each hydrogen can actually lead to the formation of a hydrogen bond or an electrostatic interaction with a negatively charged, partial negative charge in another molecule. In this case, this will be impure water, so among water molecules. Now, because of the two hydrogens, each water molecule can actually form two hydrogen bonds with each other. And this is what it implies also that you require twice as much energy now to actually break one water molecule free, which has big implication in terms of thermal capacity. And also when we'll talk about perspiration, you'll see later. Now, because of these double bonds, now water molecules, they attract each other with a lot of strength. Okay, hydrogen bonds, if you remember, they're not very strong. We're talking about billions of them in a pure water liquid. You really have a lot of force there. So the tightness through which water connect with each other in general is referred to as cohesion. Water has a very high cohesion. So these water molecules, they tend to stick together very strongly. Okay. And now this is exercise also on the surface molecule. So if this is the water molecules that are in the surface, now these water molecules are pulled down very tightly through the hydrogen bonds by the other water molecules. And also they connect with each other very tightly through the hydrogen bonds. So this gives origin to what is called the surface tension, which is something that force exerted by these molecules here. You can see, for example, that prevents, for example, this spider from going through the water. If you think, you would expect this spider, which has a higher density on the water, to go beneath. But the force that each of these legs is exercising on this water is not strong enough to break all of this strong connection of these water molecules. That's the same reason why you, for example, different tents, they are waterproof in a sense that they can withstand water when water forms droplets because of the surface tension and the cohesiveness. But then if you squeeze actually one of these drops, what you will see is that you break the surface tension, the water molecules start spreading, the water actually go through. How is it important this in biological system? Here see an example. Plants, they actually exploit this cohesiveness and this surface tension. So what happens in plants? If you think, plants have to move, like this tree can be several meters high, and it still has to move nutrients from the ground all the way to the leaves. However, I don't know if you ever noticed, as much as we like to hug trees, trees do not have a heart. They don't have a pump. We use a pump to move blood around. Trees don't have it. How do they move this liquid in that direction? What they do, they exactly use this cohesiveness of water. So how do they do that? Now, trees have a large surface here, huge surface on the top made by the leaves. The leaves, they tend to lose some water molecules. They tend to have evaporation taking place. Now, what happens when evaporation takes place? You have that the water molecules that were originally on the surface, now they're replaced by other molecules that were underneath. When they do that, because of the hydrogen bonds and the cohesiveness of water, this leads to the pulling up of all the other water molecules that were underneath. So now the evaporation of the water creates a movement of water molecules towards the surface, which is exactly what's happening here. And now this translates into the movement overall of all. This is together with other factors. I want to specify that, like osmosis and other factors there. But this leads ultimately to the movement of water in the direction from the bottom to the top. So the cohesiveness is used to pull all these water molecules and all the nutrients that are found mixed in the water here upward all the way to the leaves. Another huge property of water is thermal capacity. What we mean by thermal capacity is the amount of energy that water needs to absorb in order to change phase, to go from liquid, for example, to gas. Now, the range of temperature in which water is stable, if you remember, I stressed on the previous lecture, or when we're talking about origin of life, how stability was important for the origin of life. Evolution works well in stable conditions, not in ever-changing conditions. So water guarantees that stability because it has a huge temperature range in which it always stays liquid. This theoretical range is between 0 degrees, which is referred to as the melting point, where water will become solid, or from solid will go to liquid, to 100 degrees, which is referred to as the boiling point of water, where water would evaporate, or from a gas would turn into liquid. Now, because of this, if you think on Earth, considering the distance we have from the sun, we have a certain temperature range, which basically guarantees that water will always stay. This temperature range is mostly between this temperature range of water, so the temperature range we have on Earth, guarantees that water, the vast majority of water, will stay actually in the liquid state. This guarantees stability for the living organism that live in it, and at the beginning of life, assuming that life has started on water for the first organism. Now, there's another property of water which even extends this temperature range, and it's the fact that even when water freezes, so when we're reaching the melting point, when water freezes, this actually leads to the formation of permanent bonds between water molecules, which you see here on the right side here. When these bonds become basically permanent, these hydrogen bonds, and that's why you have the formation of crystals, now the spacing between molecules becomes a little bit more than what it is when the water molecules are free to move around. The water molecules are actually a little bit more dense than when they're frozen. So now, what it means is that if you have water that is exposed to temperature lower than 0 degrees, you will have formation of these crystals, but because of the low density of these crystals, they will move upward towards the surface. When they move upward and they accumulate on the surface, they start forming this ice surface on the water. Why is this important? It's because now actually you have something, the ice, that start actually preventing or start working as an insulator of the remaining liquid water underneath. Ice is an excellent insulator. That's why you make igloo, right? So what happens that now, even if the temperature on Earth goes well below 0, imagine North Canada. You would have that in a lake in northern Canada. You have temperature of -40, -50. Now, even those scenarios there, if these temperatures don't last forever, obviously, but if they last even for a few months, you have that now this ice will work as an insulator for the water underneath. You will still have liquid water in large lakes. Living organisms there, like fishes, could still survive, will still be in stable conditions. So this temperature range can even be extended by the lower density of frozen water or solid water compared to the liquid water itself. Another important property of water is that it's a consequence of the two hydrogen bonds we were talking about earlier on. So each water molecule is bound to the other through two hydrogen bonds, right? So now what it means is that each molecule has to leave the liquid surface. So when water evaporates, and evaporation takes place not only at 100 degrees, but even at 20 degrees, even now. So even on your skin right now, you have water evaporating, right? Now, when it gets hotter, you would have more water evaporating, right? You start producing more and more sweat and more water as a consequence evaporating. Now, because of the two hydrogen bonds, what you have is that each molecule that now leave your skin is actually taking twice as much energy that a molecule bound by a single hydrogen bond would take. So water is now very efficient at taking energy away through perspiration from your own body. Right? So when you are heavily sweating, it's really hot, you have a lot of water, sweat form in your hand. Now, when these molecules are leaving, they're taking energy. Each molecule is taking the energy corresponding to the hydrogen, two hydrogen bonds. And by doing that, you have the energy from your body being taken away very efficiently. And that's why you start feeling that cooling sensation. When air is blowing through your skin, moving away these water molecules, increasing the number of water molecules going away from you and taking that heat out of your own body. Finally, another great property, that's the last property of water that I'll discuss here, which makes it such an amazing molecule for life, is its capacity to work very well as a solvent on many, many biological molecules. Now, water, it's a polar molecule, so it has some partial positive and negative charge. And because of this, these partial and negative charges allow water molecules to actually form electrostatic interactions with other fully positive or negative molecules such as ions, or partly positive and negative molecules such as other hydrophilic molecules such as glucose, for example. Different proteins here, you can see they would have amino acid here that are polar on the outside, so they would have some partial charges which then can bind to the water molecule around. Ions too, for example, that's a salt put in water. Sodium and chloride get separated because the water molecules, now the partial charges of the water molecules, can form some bonds with the negative charge of chloride or the positive charge of sodium, forming what we call hydration shells. So why is this important? Because now you have a molecule can dissolve most of the biological molecules. You see, when we talk about proteins, DNA, so carbohydrates, we're talking mostly about polar molecules. Now you have a molecule that can dissolve them. Not only that, but it can move around, can be used now inside the body where it's moving around in your blood, for example. It's an aqueous solution, plus containing also proteins and so on. In plants, now you have that water, since it can dissolve so many biomolecules and can move around, can actually carry these nutrients and ions around bodies. So water can work as an amazing solvent and as an amazing vehicle for all these nutrients, polar nutrients and biological molecules. Moving to the next topic now, we'll talk about pH. So when we talk about water, if you remember, we define water as a polar molecule because it has two polar covalent bonds, which are not opposite to each other. And the reason why these bonds between oxygen and hydrogen are polar and non-ionic is because the difference in electronegativity is lower than 1.7. It's around 1.25. Now, this is true obviously means that these electrons in between hydrogen and oxygen are usually shared all the time. In rare instances, though, the high electronegativity of oxygen leads oxygen to actually take the 2 electrons that are shared here, basically leaving the hydrogen without electrons as a single proton. So this is referred to as dissociation of water. It takes place all the time, water, at very, very low rate, extremely low rate, but it happens. And now these protons that are released, in reality, they're not really free, but they're bound by other water molecules, forming what is called an hydronium ion. Sorry, here I made a mistake. There is a 3 here. It's supposed to be. Now, commonly, instead of talking about hydronium ions, we refer to as protons or hydrogen ions, as if they were free, but in reality, they're bound by other water molecules. Now, the measurement of the concentration of these hydronium ions or protons in the solution is referred to as the pH. Now, it could be measured, this concentration, in molarity, but this will give you very small numbers. For example, this is the concentration of hydrogen ions in pure water, which correspond to the dissociation of water. I say it's very low. In fact, the concentration of protons found in pure water will correspond to 10 to the minus 7 molar. It's an extremely low volume. We haven't talked about molarity yet, but take it as a very, very small volume. So it's 0.000, so on, 1 molar. Now, these are not small volumes. They're not very comfortable to work with. It's much easier if you actually work with a logarithm, because once you convert it to a logarithm, you're talking about just the exponential. Now, if you make it negative, even these negative values will become positive. And that's the reason why the concentration of protons in a solution are now expressed in pH, which corresponds to the negative logarithm of the protons. So how this converts, for example, 10 to the minus 7 will convert to a pH of 7. So now, as I say, this is the concentration of protons in the form of pH of pure water. And this is what is referred to as the neutral pH. So the middle point of the pH scale. pH scale that now can go to values lower than 7. They're referred to as acidic values. They're brought to there by molecules called acids. We'll talk about that. And they can go down all the way to 0. Or it can go higher than 7. In this case, we're talking about a basic or alkaline range, which can go all the way to the 14. And this is induced by molecules that do reduce the concentration of protons called bases. Talking about acids and base. So when it comes to acids, what we're talking about is molecules that, because of a high difference in electronegativity between one element, so oxygen, like in this case, and hydrogen, they tend to release protons, okay, or hydrogen ions or hydronium ions. Here you have an example of this group. This is called the carboxyl group. We'll talk about that in the next lecture. But just talking briefly about this structure, is made by a carbon with a double bond to an oxygen, with a polar bond here. And then this is attached to an oxygen, which is then attached to a hydrogen ion. Now, normally this bond will be a polar covalent bond, non-ionic. However, the presence of this group here makes this bond, brings this difference in electronegativity to a higher value than the theoretical 1.25, to the point that this oxygen here now, its electronegativity gets higher overall, and so tends oftentimes to take both electrons out of this. By doing that, it would end up having an additional electron compared to the number of protons, and therefore ionize negatively, becomes a negative ion here. And the hydrogen will lose these electrons and therefore would end up having only a single proton and become positively charged. This dissociation in these type of molecules makes them act as acid, and these molecules are referred to as acids. So acid, based on the Lewis definition, are all those molecules that do release protons. Now there are strong and weak acids. We won't get into that. This is not the scope of this lecture. Oppositely, there are other molecules which now instead are referred as bases. These molecules that do release a lot of protons, they would tend to bring the value of the pH. So imagine if this concentration goes up, the pH goes down. And so they bring the pH value towards the lower side of the range, so lower than 7. Bases, they have an opposite effect. They do bind protons, and by doing that, they do decrease the exponential here values and therefore increase the pH, bringing it towards the 14 values. So bases are molecules like this one with high electronegativity. So they have an element with high electronegativity like this nitrogen here in this amino group. It's a group commonly found like the carboxy group in amino acid that has high electronegativity. So it has a partial negative charge here, which then can attract positively charged molecules, such as a proton. And so by doing that, it can bind protons to these two electrons here not share. And by doing that, it removes the proton from the solution and lowers the concentration of protons, increases the pH. So molecules that do bind protons are referred to as bases, and they increase the pH up. Now, sometimes some bases are formed by the dissociation of an acid. For example, this acid here, acetic acid, as a carboxy group, is exactly this molecule right here. When it breaks down, acetic acid releases the proton, which forms an hydrogen ion. At the same time, it's forming a molecule here that now could bind, because of the negative charge, can bind actually protons, which is a base. So you can actually have both. So the acid that forms the base. This is the base of buffers. We'll talk about that in the next few slides. So why is the pH important in an organism? The pH is important because it controls in different parts of the body different phenomena. So pH is commonly used, for example, to turn on and off enzymes. So look at this example here of the pH value that you find here in different part of your gastrointestinal tract. So this is the series of organs that together basically are in charge of the digestion of the food. So going all the way from your oral cavity to the end point, which I won't mention here. So if you look at the pH, now in your oral cavity all the way to the end here of your esophagus, you are in a range which is around 6.8 to 7.5. As soon as you enter your stomach, your pH dramatically goes down to 1.5 to 2. And then it goes back up to values all the way to 7 or even 8 here in your colon. Why is this taking place? It's because there are enzymes that do work at different pH. For example, your enzyme in your stomach, they work extremely well at lower pH, 1.5 to. They don't work at all at pH 7. So by keeping different pH values now in different parts here, you would have that this enzyme will be active here, but as soon as they move into the intestine, their activity can be stopped by simply changing the pH. Similarly happens within your cells. Each of your cells, they have an organelle called lysosomes, which are packed with enzymes that work very well in acidic conditions. These acidic conditions are present inside the lysosome, but not in your cytoplasms. So that if this lysosome bursts, the enzymes are released in an environment which now has a pH much higher and turns them off, so they don't degrade the cell. Now, how is the activity affected by the pH? Here you have an example of how tight the pH has to be in different parts of your bodies and how important it is. For example, in your blood, your pH is around 7.4. This is the range which is still okay, between 7.35 and 7.45. But you can see how small changes overall. So for example, if your pH goes all the way down to 7, it's not a huge change, but it's dramatic for your body. You get a condition called acute respiratory acidosis. This happens usually if you, for example, you're in an accident, you stop breathing or you choke on something. You can't breathe anymore. You stop breathing. This causes accumulation of carbon dioxide, which your red blood cells turn into an acid, carbonic acid, and therefore brings the pH low. Now, if this condition is not treated quickly, your brain will shut down. You get into a coma, and all the functions controlled by your brain will stop too, ultimately leading to death. The opposite, if your pH goes higher, which is a condition called alkalosis, caused by different situations, like for example, hyperventilation, these change, which is again, is not a dramatic change to 7.8. We're not talking about pH of 10, 7.8. If your blood gets to 7.8, now you start having tetany. Tetany is the uncontrolled spasm of your muscle. So keep getting this signal. This includes also your muscles in your lungs, in your digestive system, meaning your digestion will stop, your breathing will stop. You can see how if it doesn't get treated, you would die fairly quickly as well, by alkalosis as well. How do organisms prevent all these changes, this swinging of pH, so they keep the pH stable? They do that by using buffers. Buffers are systems which consist of two components. One thing called weak acid and the conjugate base or weak base associated with that. And these two components, they turn into each other depending on the pH or the changes in concentration of hydrogen in the system. Again, going back to the example of your blood pH, it's maintained within this range, 7.35 to 7.45. How this is done is by using what is called a carbonate buffer, which consists of carbonic acid, this molecule here, which is the weak acid, and the base that is formed when carbonic acid loses protons, one proton, which is bicarbonate, this molecule right here, which works as a weak base. Now, when you have both of these molecules present in your blood, you're in a situation in which you can control both an increase on protons and a decrease in protons by having one of the two molecules converting into another one. Let's look at this example. So imagine you are in your blood here, and you are in a scenario where your blood, for example, you stop breathing and you start accumulating a lot of acid there. If the buffer wasn't there, the concentration of protons will go crazy high, your pH will go extremely low. But now, if you have this buffer here, what happens is that all these excess protons that are introduced there, they can actually bind to the weak base here, bicarbonate here, which then, by binding a proton here, convert into the acid form, carbonic acid. So now you would have the amount of bicarbonate will go down, carbonic acid would go up, but ultimately the concentration of protons would have been kept the same because all the excess of protons would have been removed by this conversion into carbonic acid. The opposite happens if you have a base that is introduced in your blood. At this point, the proton's concentration will go down. This would lead to an increase in pH, but this is compensated by more carbonic acid converted into bicarbonate. And when it does that, it releases protons, which then compensate for those protons that had the base. By doing that, the concentration of protons will be kept stable, and therefore the pH will be kept stable. Obviously, buffers like this one will work only when both forms are present there. So imagine if you're running out of, for example, of bicarbonate here, so this is not present anymore. You introduce a lot of protons, there is nothing to bind these protons and convert into carbonic acid. So the buffer would not work. So that's how living organisms, they prevent spikes in pH in different organs and organelles or cytoplasm and so on by using buffer. There are many types of organic buffer. There are phosphate buffers inside your cells, for example, other than this carbonic acid. So there are different type of buffers, and some of them are actually used also in the lab to control the pH, even in solutions external from your own body. So in summary, what we talked about here, we talked about why is water such an important molecule for living organism. And we talked about how basically all the properties, the thermal capacity, the cohesiveness, and the capacity of dissolving lots of biological molecules, they all arise from the polarity of water. Then we talked about pH. We defined what it is, and we defined acids and bases and how they affect the pH. And then we talk about why is pH important in living organism? How is controlling the activity of different enzymes in different organs? And finally, we finish up by talking about how living organisms, they do control the pH in their different compartments by using buffers that can bind excess of protons or release protons whenever it's needed.